welcome everybody.
Sorry,
Welcome everybody. It's great to see such a great turnout to support Chris, and not just to support him, but to hear about the really incredible work he has done over the last 5 years or so. I feel really fortunate to have had Chris working in the lab on some really exciting experiments, I think, over the last number of years. We've been through lots together over 5 years, and you'll see, I think, the fruits of his labor in this. One of the things maybe I'll say about Chris is, most of the students that we work with in this area come from have their primary background in education is in engineering. Chris's background originally was in neuroscience, which is a little different. He did a master's in a master's program in bioengineering. But I think one of the things you'll see throughout this presentation, and has been evident in the work he's done throughout the years, is he's really brought that neuroscientific way of thinking to a lot of the work that we've done, which often has a fairly engineering heavy focus to it. So Chris, one way to maybe summarize the difference between a lot of things is, engineers, when there's a problem, the first thing they want to do is, if you're a mechanical engineer, you open up SolidWorks and start trying to design a widget to solve your problem. If you're if you're a computer engineer or computer scientist, you try and solve all your problems by writing new code. Chris would always go towards mechanism, like how does the brain, what's the brain doing? And I've really valued that perspective and that input and insight that he's brought to our lab over the last number of years. So I won't belabor the point, Chris. I'm really pleased that we're here. I'm really excited to see, well, I've seen it before. I'm excited to see it again. And I think everybody's in for a treat. He's done some awesome work. So Chris, why don't you take it away?
Cool. Thanks, Rob. Let me go ahead and share my screen and get this PowerPoint going. So as Rob mentioned, my name is Chris, in case you didn't already know that. But today I'm going to be talking to you about my graduate work that I've done in the rehab neuroengineering labs with Rob, really studying how we can use intracortical microstimulation in humans in the somatosensory cortex to restore touch for a brain-computer interface. And you can see I have Biomimetic in the title here. There's going to be a lot of different work I talk about and kind of all united under this framework of biomimetic stimulation. If you don't have time to ask your questions during the Q&A or anything, feel free to email me. My email's here and will be at the end of the presentation as well. So first of all, for people who maybe aren't as familiar, what is a brain-computer interface? So a brain-computer interface can refer to really any device which interfaces with the brain to restore function to people with neurological injury or disease. Typically, when we talk about brain-computer interfaces, we're talking about these implanted intracortical electrodes in the motor cortex, which we can use to decode activity for the control of a robotic arm or other end effector. And so on the right here, I have a couple of examples of sort of historic work in this area from both BrainGate and our lab, where there are these people, again, who have some sort of neurological injury or disease implanted with microelectrode arrays in the brain, and they're able to control a robotic arm for self-feeding. But we can also do some fun things with BCIs. And so here's a video of me having a lightsaber battle with our current participant, P2. And this is on May the 4th, which is his implant day. And you can see I'm controlling the lightsaber with my own hand, but our participant's controlling the lightsaber with a robotic arm. And there's actually 4 sensors on that hand. And so he could detect the lightsaber in his hand. And of course, in becoming a Jedi Master, you need to be able to detect the Force. And I apologize for people who've heard that pun before, but it's one of my favorites. So, but even if you're not a Jedi Master, being able to detect the Force and get this sort of sensory feedback is important for normal motor control. So here you'll see someone try to light a matchstick on the left, and they have normal intact sensation, and they're able to do it in just a few seconds. But on the right, they have their fingers anesthetized so that they have normal motor control. But they're lacking that sensory feedback. And you'll see when they're lacking this feedback, it takes them much longer to light the match, even though eventually they're able to get it. And so the goal of our work really is to not just restore motor control, but also to restore sensory feedback. So we have microelectrode arrays implanted in the motor cortex, which we can decode activity from for the control of a robotic arm, but we can also stimulate through electrodes in the somatosensory cortex to restore touch via that arm. And recently, we demonstrated that sensory feedback can actually improve motor control. And this is from a paper published in Science with Charlene as first author, and I'm a co-author on it. And you'll see on the left, the participants controlling the robot arm with feedback, and on the right, they're controlling it without feedback. And for the fastest transfer times of these tasks, you'll see that he's always better with stimulation. And this is really cool and really interesting, partially because this is very simple feedback that we're providing. We're providing sensory feedback on a single electrode with linear encoding of force. And even with this very simple feedback, we can see improvement in motor control and in transfer time. But intracortical microstimulation in the somatosensory cortex in humans is very new. Our participant is actually the first person with these kinds of implants, and that was just in 2015. So this image here is from Charlene's sort of seminal paper published in 2016, where they demonstrated that you can use these implanted electrodes to drive sensation in the upper palmar region of the hand. And these follow sort of a general somatotopic organization. But there's a lot that's not understood about ICMS. So you can evoke these different intensities on different electrodes. You can also evoke these different qualities like tingle, pressure, and warm. And these happen on sort of electrodes in a seemingly random basis. And it's not understood why this occurs and how we could potentially control it in terms of the stimulated parameters. So again, ICMS is kind of this very new thing, not very well understood. And so the goal of my graduate work was really to better understand how we can use ICMS to improve sensory feedback for clinical applications. And so I divided this goal into really three overarching goals. The first is to investigate how well stimulated electrodes hold up over time. And this is because if these electrodes aren't able to record or stimulate over long periods of time, then they're not going to be practical for these clinical applications. Then I also want to understand how we can modulate parameters of ICMS in a way that modulates perception. And this is really, again, based on that previous lack of information we have about how these things can affect quality and intensity. And then finally, I wanted to use these things that we learned about the parameters to really develop better methods for sensory feedback. Before I jump into those goals, I'm going to discuss with you some of the general methods that will be applicable through all the experiments that I did. So we have two participants at the University of Pittsburgh. The first is P2, who was implanted at the age of 28 in 2015, and they're still implanted to this day. So it's over 6 years now, and they're still going strong. And we have participant P3, who was implanted also at the age of 28, but in 2020. So they've been implanted for less than a year. And both of these participants are stimulated with these Utah microelectrode arrays. Both have 2 arrays in the motor cortex and 2 in the somatosensory cortex. Importantly, the motor cortex arrays are 10 by 10 with almost every electrode wired, whereas the somatosensory arrays are 6 by 10 with every other electrode wired. And this was due to wiring constraints. Another important difference actually between the 2 participants is in P2, these motor arrays have platinum electrodes. The somatosensory arrays are iridium oxide. In P3, all of the electrode arrays are iridium oxide. So I'll bring that up again later when we're talking about stability. So when I talk about electrical stimulation of the brain, you might imagine something like this. So something very general and kind of unsafe. But actually what we're doing is much more focal and safe. So here is an image of one of those electrode arrays that I showed you earlier. But this is an explanted array, so this is why one of these electrodes here is broken. But this is what they look like. And again, every other electrode is wired, so we can only record and stimulate on every other somatosensory electrode. And when we do stimulate on those electrodes, this is what our stimulation protocol looks like. We stimulate with these biphasic charge balance pulses that have a 200 microsecond cathodic phase, a 100 microsecond interphase, and a 400 microsecond anodic phase. And there are really 3 primary parameters I'll talk about that we vary with these experiments, which are amplitude, frequency, and train duration. So amplitude refers to the amount of charge injected in this cathodic phase. Frequency is the number of pulses per second, and train duration is just the total time that we stimulate for. And all this stimulation is delivered through a Serostim R96 stimulator provided by BlackRock. So now I want to describe briefly, I've mentioned this term biomimetic. I want to tell you what that means and put it in the context of a more engineered approach. And so I'm going to use some examples from peripheral nerve stimulation. So this was work done at EPFL under Silvestre Machera and Stani Raspopovic. So this first paper from 2014 shows what we'd consider more of an engineered approach. So they had these sensor readings that came from a robotic hand, and they transformed that directly into stimulation current. So there's a sort of arbitrary mapping between the force readings and the stimulation current. And this is also what was done in that science paper that I showed you earlier. And so this is really a more engineered approach. It's kind of arbitrary. Whereas a biomimetic approach aims to put the stimulation more in the context of real neural activity. So this is from that same group from 2018, where they used the biomimetic approach. So you can see there's this sort of mechanical indentation profile, and they stimulate with higher currents during the onset of this profile, and then lower currents during the maintained contact. And this is really what normal peripheral nerve activity looks more like, as Sliman Benzemaya's lab has shown previously. And they've also shown more recently that the cortical activity also looks like this. So this is from Terry Collier's paper published in 2019, where they provided mechanical indentation profiles to non-human primates and looked at the cortical activity, and they found increases in both the area of activation and the rate during these sort of transient contact events. And so when we talk about biomimetic stimulation, we're primarily talking about modulating stimulation based on these contact transients rather than this sort of arbitrary mapping of stimulation. So now that I've described to you those more general methods, I'm going to jump into these, the first of these three goals, which is investigating how well stimulated electrodes hold up over time. So there's been a lot of studies on this stuff in animal models, and particularly non-human primates, but I'm going to talk about just one paper today that gives a really comprehensive overview, Baresse et al. 2013. And this is a really nice illustration from that paper where they show the various ways in which electrodes can fail over time. So there are biological failures, which include neuronal death, gliosis, bleeding, and a meningeal encapsulation. There's material failures, such as the electrode tip breaking, the insulation degrading over time. And then there's more mechanical failures, which happens on the connector side, so things like wire damage. And so as you can see, there's really lots of ways in which these electrodes can fail over time. And if you look at non-human primates, actually over half of arrays fail within the first year with a median time to failure of 182 days. And this is not super promising for clinical applications because the surgery that's required for this is highly invasive. And so if these things can only last for less than a year, then they're not going to be useful. But I think an important thing to note is in these non-human primate studies, about half of these failures occurred due to mechanical failures, and particularly the connector. And if people who work with monkeys know, monkeys tend to sort of fiddle around with their pedestals. So this might not be directly translatable to humans because humans don't tend to monkey around with their pedestals. So, but even when these electrodes don't fail, you do see decreases in signal over time. So this is assessed here at the mean number of viable channels, which are channels that are able to record high amplitude recordings. And you can see even when they don't fail, these high amplitude recordings decrease over time. And an important distinction between what they showed in this paper and what we do in our work is we're often stimulating on these somatosensory electrodes, which is known from animal studies can cause further material damage. So we wanted to know if stimulating can actually cause further degradation of the signal or if it would be unchanged. And I've been talking about sort of low and high amplitude signal, and I kind of just want to show you a hard example of what that means. So here on the left, you see what we consider a low amplitude signal. These are recordings that basically barely cross threshold. It doesn't have that really distinct neural waveform that you typically associate with recordings, and it's low amplitude. And this could be due to sort of multi-unit activity far from the electrode or noise at the tissue interface. But this one on the right is a high amplitude signal, and so you see this sort of classic shape of this neural waveform. You see it's large in amplitude and well isolated. And so I'm going to talk to you about different parameters we use to assess these waveforms, including the peak-to-peak voltage, which is just the distance between the trough and the peak of the waveform, the noise, which is sort of the deviation in this baseline, and the signal-to-noise ratio is just a function of these two things together. And then the high amplitude units are any neural waveforms that have an amplitude over 100 microvolts. And you can see this one, based on this axis on the left, would be considered a high amplitude waveform. And so now I'm going to present to you results from a paper that's currently on medRxiv and revision at Journal of Neural Engineering, where we looked at how the non-stimulated motor electrodes compared to the stimulated somatosensory electrodes in P2. And what we found was at the beginning of implant, the non-stimulated motor electrodes had a lot more of these electrodes with high amplitude recordings. And this is kind of expected for two reasons. One, we just have more motor electrodes. But the other reason is that the motor cortex has these large BET cells, which can produce high amplitude waveforms. And so this wasn't surprising that they were different. But what was kind of surprising is if you look at post-implant day 1500, now they're much closer together. And so if you look at this as a representation of the percent of electrodes with high amplitude units, you can see that the non-stimulated motor cortex arrays are decreasing more over time than the stimulated somatosensory rays. So this could mean that the stimulation is actually allowing us to record more high amplitude waveforms over time. But there are also, again, another important distinction between these two rays is one is platinum, one's iridium oxide, and also they're implanted in different areas of cortex. So at least what we're seeing here is stimulation doesn't seem to be causing further degradation of the signal, but it might actually even be helping a little bit. And then when we look at the peak-to-peak amplitude, noise, and signal-to-noise ratio, you see that the motor electrodes, the non-stimulated motor electrodes, and the stimulated somatosensory electrodes are behaving pretty similarly over time. So again, it seems that stimulation isn't causing further degradation of these trends, although you do see decreases on all electrodes over time. And then we also want to know, so we want to know if we could record on these electrodes over time, but we also want to know if we can continue to evoke sensations at low amplitudes. And so we tested this using a detection task where the participant would receive stimulation on a single electrode on one interval, and then they would not receive stimulation on the other interval. And this would be sort of randomly ordered. And the participant would have to indicate which interval contained the stimulation, 1 or 2. And then we could use this to determine a threshold. Basically, if they got it correct, we would lower the amplitude. If they got it incorrect, we would raise the amplitude until we were able to determine a detection threshold. And what we found was at day post-implant 100, the median threshold was around 32 microamps. But then by day post-implant 500, this is actually decreased to below 20 microamps. And this was a significant difference. And then at days 1000 and 1500, you could see this remained pretty stable and maybe even decreased slightly. So this is really promising because it means not only are we able to evoke sensations over long periods of time, but we're actually able to evoke them at lower amplitudes. So his ability to detect stimulation only got better over time. And so this means we're able to continually stimulate at low amplitudes over long periods of time, which is probably one of the most promising results for clinical implementation. Next, I want to talk to you about some material stability results. And so this was done in collaboration with Tracy Shui's lab. And this is a manuscript that's currently in preparation with Kevin Weppel as first author and me as second author. And we looked at these explanted electrodes that were implanted in 2 different participants, but I'm only going to focus on a couple of those electrode arrays that were implanted in P2 that received a brief amount of stimulation. And what we found was even a small amount of stimulation can sometimes cause damage. So there were 2 stimulated arrays. This one on the left here, you can see the white arrows are pointing at these electrode tips that showed some sort of demetallation. And this was caused by stimulation, which you can tell because there was a sort of checkerboard pattern which corresponded to the wiring of the arrays. Whereas in the other array, we didn't see any damage as a result of stimulation. And so I wanted to know kind of if there was any predictors of this, what was causing this damage. And what I found was that increased charge injection only at more negative voltages was correlated with damage. And so it seems that even with small amounts of current, because these were implants that are only implanted for 180 days, even with small amounts of stimulation, you can get this sort of damage on the electrodes. But what was really interesting from this was when we then went to correlate damage with signal quality, we found that there was actually no significant correlation. So both the damaged and undamaged electrodes were able to continue to record signal. And there's a couple of caveats to this, a couple of reasons why this analysis was a bit difficult, which I could explain later if people have questions. But basically, this seems to indicate that even when we do damage electrodes, we're still able to record from them, which is kind of an interesting and maybe unexpected result. So conclusions from this whole first goal from all this analysis on stability is that the Utah arrays are able to record and stimulate for many years, although signal on all electrodes still declines over time. Stimulation doesn't seem to cause further decreases in the signal, and maybe even in terms of high amplitude waveforms can improve our ability to record them. And then detection, this is probably the most promising result, detection thresholds only ever improved over time, meaning we can continue to stimulate for many years. And again, our current participant P2, or one of our current participants, has now been implanted for over 6 years, and we're still able to evoke sensations. And finally, we found that a small amount of stimulation under certain conditions can cause electrode material damage, but this may not actually result in an inability to record or stimulate. And finally, to draw this back into the context of biomimetic stimulation, so even though we didn't see any degradation in our ability to record or stimulate over time, we did see that some material damage could occur when you provide more charge, and particularly more charge at more negative voltages. And biomimetic stimulation can actually potentially then improve electrode longevity or material integrity by delivering less charge over time, because you're only stimulating during those contact transients, which results in less overall charge injection. So potentially biomimetic stimulation then provides a solution to this material damage. So now I'm going to jump into the second goal of my graduate work, talking about how these stimulus parameters actually affect evoked perception. And the first task I'll talk to you about is really based on the intensity of evoked percept. So we use a magnitude estimation task where we'd stimulate on a single electrode with some set of parameters, and then the participant would report the intensity on a self-selected scale, so something like 1.5. Then we would change the parameters and stimulate on that same electrode. So here, for example, we're increasing amplitude, and the participant might report something else like 3. And this is a self-selected scale, so theoretically the participants could report anything, but typically they would report values from 0 to 6. I think this kind of experiment, in contrast to the detection experiment, really emphasizes the importance of human experiments, because the humans can tell you directly what they're feeling. Whereas if you try to do this in a macaque monkey, for example, you stimulate it on a single electrode and ask them to report the magnitude, they might say something like ooh, ooh, ah, ah, and that's just objectively less informative than numerical values. Of course, animal experiments have their own importance, and there's a lot of things you can do in animals that you can't do in humans, but for perception, I think humans are sort of keen for these kinds of experiments. And what we found in our first participant, P2, is as you increase the amplitude, you got these linear increases in intensity, and this was consistent across electrodes. And as you increase train duration, you got these more logarithmic changes in intensity where they leveled out after about 1 second. And so these results were very consistent across electrodes, and they were also kind of expected from previous literature in animals. But then we found these really kind of unexpected and interesting effects of frequency, where some electrodes had the highest intensity at the highest frequencies, some electrodes had the highest, but other electrodes had the highest intensities at these lower or intermediate frequencies. And this wasn't really expected from animal studies. It was expected that higher frequencies should always lead to higher intensities. And we then classified electrodes into 3 groups based on k-means clustering, and we defined these as high frequency preferring, intermediate frequency preferring, or low frequency preferring, just based on where the maximal intensity occurred between 20, 100, and 300 hertz. And we also wanted to know how these different electrode groupings and how the parameters affected the evoked qualities. And so we would stimulate on individual electrodes, and we'd ask the participant to report their experience. And so they might say something like, it felt like pressure with tingle. And this was very open-ended, so they could say all kinds of things. But you could see there's really 2 important qualities here, pressure and tingle. But again, they could say something much longer and much more vague. And so I wrote code basically that would go through these perceptual reports and extract the important quality information. And what we found was really interesting. The frequency of stimulation did impact the evoked quality, but also these groups that they were divided into affected the evoked quality. So you can see here with the stimulation frequency of 20 hertz, you got these sensations of tapping, sparkle, and touch, but those only occurred on the low frequency preferring and intermediate frequency preferring electrodes, whereas these high frequency preferring electrodes never evoked really much quality at all or much sensation at all at 20 hertz. And then as you move to 100 and 300 hertz, you see these intermediate frequency preferring electrodes are evoking these sensations of buzzing, vibration, sharp, and warm, which are not evoked on these low frequency preferring electrodes. And also the high frequency preferring electrodes are starting to evoke more sensations in general, and particularly these sensations of touch and poke or prick that were not elicited on the other groups. And so we see that the frequency is able to impact the quality, but it impacts it also in this electrode specific way that seems to be really entangled with those intensity measurements we made before. So both the intensity and the quality are behaving in this sort of electrode specific way. And we found in P2 that these kinds of relationships seem to be clustered in the cortex, where electrodes of one type of frequency preference were more likely to be next to other electrodes of the same kind of frequency preference. And you see this really clearly on this lateral array, where all these intermediate frequency preferring electrodes are next to each other, and all these high frequency preferring electrodes are next to each other. And we think that this has to do with the underlying somatosensory organization, which I'll describe in a couple slides. But first, I want to show you some results from P3, our second participant implanted with these implants. And we found similar but different relationships. So in this participant, we did find these electrodes that had these low frequency preferences where the highest intensity was at the lowest frequency, and this decreased as you increase the frequency. But the k-means clustering analysis showed us that we were better dividing it into 2 groups instead of 3 groups like we did in P2. But also you do see some sort of spatial clustering in this participant as well, but it sort of manifests differently. So in the other participant, we saw these clustered within arrays. In this participant, we see that these low frequency preferring electrodes really only occurred on one of the arrays. And so you get more of this clustering across arrays. So what this seems to tell us is that it's likely these kinds of relationships could occur in multiple participants, but the way they manifest might be affected by a lot of other factors and might look a bit different. So we're going to need to look at other people with these kinds of experiments to really determine that. But as I mentioned, we think that the spatial clustering has to do with the underlying spatial organization of somatosensory cortex. And there's a lot of previous studies done in animals where they use different frequencies of mechanical input provided to the periphery and measure the preferential activation in the cortex. And so on the left here, there is a study where they did optical imaging in non-human primates. They provided different frequencies of indentation to their palms and then measured the activation in cortex. And they found this sort of pinwheel type organization where different subpopulations in the cortex seems to have a preferred frequency. And they actually put this in the context of mechanoreceptors that have these same sort of preferential frequency ranges. But I'm going to argue in a couple seconds that I actually don't think it has to do with the mechanoreceptors. But first I'll talk about this other study, the more recent study done here on the right, where they actually found individual neurons in mouse somatosensory cortex. Using 2-photon microscopy, they found that they can have preferential frequency responses. So even not just populations, but individual neurons can have these preferential frequency responses. And again, a lot of the previous literature, including historic work from Mountcastle and Sir, kind of put forward this idea that there was submodality separation in the cortex, or these different mechanoreceptors impinge on parts of the cortex, and that's why you see these sort of preferential responses, which made sense because they're providing mechanical input to mechanoreceptors. But actually in our results, what we're doing is we're bypassing mechanoreceptors entirely and stimulating directly in the cortex. And so I think what really is going on here is not that you get preferential responses to mechanoreceptors, but rather you have some sort of preferential organization in the somatosensory cortex for extraction of these different tactile features. And this is something more so that this more recent paper is making the claim for. I think also aligns well with some of the literature, particularly literature from Slimane Benzemaya, sort of making the claim that the cortex is really receiving a lot of convergent input from mechanoreceptors, so it's unlikely that you have this sort of label line method. So again, I think our results support more so the idea that you're actually getting preferential responses in the cortex based on tactile features such as frequency or even quality like we saw in our results. And so the conclusions from this work are that amplitude and train duration have these consistent effects on intensity, whereas frequency had these really interesting sort of electrode-specific effects on intensity and quality, and these things cluster spatially, which we think has to do with the underlying somatosensory organization, although there are other alternative explanations as well for this. And finally, again, to bring this back into the context of biomimetic stimulation, if we're really truly trying to provide biomimetic stimulation, then we're likely going to have to provide frequency modulation on a more electrode-specific basis based on this underlying somatosensory organization. And you'll see in my next bit of work on biomimetic stimulation that this seems to hold true. So now moving into the final goal of my graduate work, talking really about using this knowledge that I developed on parameters to develop better methods. And again, I'm going to be using this sort of biomimetic approach. And so the way I approached biomimetic stimulation to begin with was we took non-human primate data that was collected in Slimane Benzemaya's lab while the monkeys received tactile indentation of the hand. And I built 2 pulse trains from this. The first is this sort of biomimetic timing train where we looked at a single recorded unit and we matched the timing of the pulses to the timing of the action potentials from that recorded unit. And so we were modulating the frequency content, but maintaining the amplitude as constant. And the other train, it's a biomimetic amplitude train. We looked at this sort of multi-unit population level activity and z-scored the spike rates to modulate the amplitude based on the population response. And you see you get this sort of characteristic onset and offset activity that I talked about previously. So again, we have 2 trains, one that has a fixed amplitude but modulated frequency, and one that has a fixed frequency but a modulated amplitude. And we expect these things to actually affect neural recruitment differently, where the biomimetic timing train is more likely to recruit the same neurons based on that fixed amplitude, but each of those neurons are sort of experiencing this different biomimetic profile where they have higher frequencies at the onset and higher frequencies at the offset of contact, whereas the biomimetic amplitude modulated trains are going to recruit different neurons throughout the train because of the changing amplitude. So for example, neurons 1 and 2 would experience continuous stimulation, whereas neurons 9 and 10 would only experience stimulation during those transients. So this is sort of 2 different methods for biomimetic modulation, One, you're modulating really the rate of individual neurons, and the other, you're modulating the area of cortex activated. And we want to know if either of these biomimetic approaches could improve the naturalness and intuitiveness of the evoked percepts. And the way we tested that for naturalness was to use a 2 alternative force choice test, where we would stimulate on an individual electrode with a set of fixed parameters, so fixed amplitude and fixed frequency. And then on the other interval, we'd stimulate with a biomimetic waveform, and we had randomly ordered these two.
On Saturday. Oh, I thought it was today. Yeah.
Okay, sorry about that. Anyway, so we had these biomimetic waveforms, and basically we're asking if they're more natural. So we play a standard train and then a biomimetic train, and the participant would indicate which interval contained the more natural stimulation. And we define naturalness pretty straightforward here to mean something you could experience in everyday life with normal tactile input. And this naturalness question is kind of a subjective one, which I'll talk about a little bit later. And we kind of followed this up with other experiments. So first of all, we wanted to know if these biomimetic timing trains can increase naturalness. And so we compared it again to these fixed parameter trains. And here the x-axis represents different electrode numbers or different tested electrodes. And the y-axis represents the different parameters we tested. And you can see when we first tested against these fixed 100 hertz trains, the biomimetic timing train was generally leading to more natural sensations. So blue here indicates that the participant was choosing the biomimetic train as more natural, and the asterisks indicate that this was a significant difference. But we knew that frequency itself could impact the quality and naturalness of the evoked percept. And so we also want to test this against 20 hertz, a low frequency, and 250 hertz, a high frequency. And we found when we tested against these frequencies that the results were much less consistent. You could see for the 20 hertz train, he's often choosing the fixed parameter comparison. And for 250 hertz, there's often not a significant difference. And so overall, you see that he's choosing the biomimetic trains in 11 of these comparisons. He's choosing the fixed parameter trains in 11, and he's choosing them not different in a total of 14. And so what we conclude from this, is that the biomimetic trains do not generally improve the naturalness, but rather it seems that they have these more electrode-specific effects, which maybe isn't what we hoped for, but it also isn't surprising based on the previous work I shared with you, where we found that frequency itself could have these electrode-specific effects. And so again, I think this ties back to that point. The likely we're going to need to modulate frequency in a more electrode-specific basis to really restore natural feedback. But then we tested these biomimetic amplitude modulated trains, and we found a much better effect. So you can see there's more blue here, indicating the participant is choosing the biomimetic train more often. We also compared these biomimetic amplitude trains back to the biomimetic timing trains and saw similarly strong effects. However, you could still see there's some caveats here. So the electrodes on the far right, the 4 electrodes, you can see he's almost always choosing the fixed parameter comparison. And I'll explain in a couple of seconds of why this was the case. But first, I want to show you a report from when he was choosing these biomimetic amplitude trains as more natural. You could see the report basically is that the biomimetic train felt shorter and more like a poke, whereas the comparison train felt more like a normal tingle. And so to our participant P2, pokes felt more natural than tingle. And maybe this isn't surprising just based on the descriptors. But then when we look at these 4 electrodes on the right, where he's choosing the comparison train, he said that they both were a faint warm tingle, and he just selected the one that was more intense. And this is a bit of a confound in the experiment because we really were trying to get at this aspect of quality. But here you can see the intensity is biasing his selection. And we did some follow-up experiments I won't have time to talk about, where we actually matched the intensity prior to the experiments. And when we did that, we actually saw more robust results, where he was even more often choosing these biomimetic amplitude trains as more natural. But even without the intensity matching, we still see a stronger effect, where 21 of these comparisons were chosen as more natural with biomimetic stimulation, 15 were chosen for the fixed comparisons, and 4 were not significantly different. And so we conclude from this the biomimetic amplitude trains increase the naturalness more generally, but there were also a couple of caveats to this, including this intensity confound. Next, I want to show you some survey data. So surveys are basically where we would go through and we'd stimulate on each electrode, and the participant would have a tablet in front of them, and they would use that tablet to indicate the perceived location and projected field size, as well as the quality and naturalness of the sensations evoked. And I wanted to look at how biomimetic stimulation then compared to fixed parameter stimulation. And so I did 3 surveys with the biomimetic amplitude train and paired those with 3 surveys of the fixed parameter 60 microamp 100 hertz trains. And what we found is when we compared the natural there was a significant difference. So you could see this because this blue histogram, which represents biomimetic stimulation, is shifted to the right. But you can see the histograms are actually largely overlapping. And so we're not getting this huge increase in naturalness. We're not going from 0 to 10 or completely unnatural to completely natural. Rather, we're going from a median of 5.2 to 5.4. So again, there was a significant increase as a result of biomimetic stimulation, but it was just a small effect size overall. And so you'd expect in the discrimination tasks I showed you previously for there to be a difference, but again, it's not a huge effect size. And then when we look at the qualities that are evoked by the stimulation, you can see maybe surprisingly, they're largely overlapping. There's a lot of tingle and pressure and warm that's associated with both of these trains. But you can see for the biomimetic train, there's just a much larger amount of these poke or prick-like sensations. And in fact, they increased from 3% for the fixed parameter to 15% for biomimetic stimulation. And so it seems that biomimetic stimulation in this context is evoking the sensations of poke that tend to feel more natural. Finally, I think one of the most interesting things that I discovered from the survey data was how it changed projected field size. And the reason this is really interesting is because in the past, when we've looked at how amplitude impacts projected field size, we've never really found consistent results. But the biomimetic stimulation seemed to very consistently evoke these more focal percepts. And so what you saw was the median projected field size decreased by half. And here I showed a couple example electrodes where you saw these large changes. So on the left, you could see the fixed parameter stimulation, this green electrodes evoking these large circles, whereas you move to the right, they're consistently smaller across all three surveys. And then this blue one's even more interesting because not only are the projected field sizes smaller, but they're more concentrated. So sometimes with the fixed parameter stimulation, you would evoke sensations in adjacent projected fields like the index, tips, and thumb. But with the biomimetic, it was always in that P2 region. So I think this is a really interesting result and something that should be investigated further because it might be, and in fact, I reported previously, the naturalness increased because these things tended to feel more focal and more like a poke, and this kind of supports that idea. So it's possible that even beyond this naturalness effect, we're seeing the biomimetic amplitude modulation can reduce the projected field size and create more focal percepts. Finally, again, the naturalness question I talked about earlier is a bit subjective. And so we wanted to put the ICMS in the context of real mechanical stimulation. And so I built these mechanical force profiles with different amplitudes and rates. And we sort of used two approaches, the engineered approach and the biomimetic approach, to create ICMS pulse trains. So the engineered approach is really, again, just taking these force profiles and translating them directly into ICMS. And then the biomimetic approach was using the software TouchMime that was built in Simon Benzemaya's lab, where you get simulated neural activity based on different force profiles. And you can see, again, you get this sort of characteristic onset and offset response at these higher amplitudes and rates. And so we play a mechanical indentation and then play the two ICMS trains in random order and have the participant select the interval that contained the sensation that felt more like the mechanical input. What we found was these TouchMime trains seem to perform better overall, particularly at the higher amplitudes and rates. You can see there's a significant difference between where he's selecting the TouchMime trains more often. And the reason I think that it's really significant at these higher amplitudes and rates is if you actually look at the trains themselves, built with both TouchMime and linear encoding, these high amplitude ones are significantly different where you get these very large onset and offset transients. But at these lower amplitudes and rates, they look very similar. So if you look at this blue trace here, it looks similar between the linear and TouchMime encoded trains. So this might indicate that at these lower amplitudes and rates, we're not going to expect biomimetic to actually make a huge difference, but it's really at these more intense contact events that you're going to see a big difference in the intuitiveness. And so to summarize what I talked about in this last set of experiments, we found that these biomimetic timing trains seem to have more electrode-specific effects and didn't generally improve the naturalness, whereas these biomimetic amplitude modulated trains did generally improve the naturalness, or at least more generally with a few caveats. And they produce these more focal poke-like sensations, which are more natural to the participant. And finally, this amplitude modulation evoked sensations that more intuitively felt like the mechanical input provided to the palm. And so here are the three goals of my graduate work here summarized. And basically what I found is these Utah arrays are surprisingly robust over time for both recording and stimulating, which is really promising for our applications and for future clinical applications. And then in terms of the stimulation parameters, we found the amplitude and train duration increases intensity, but frequency had these very interesting electrode-specific effects on both intensity and quality that was clustered spatially. And again, we believe has to do with the underlying somatosensory organization. And finally, by modulating the stimulation in a biomimetic fashion, we're able to improve the naturalness, intuitiveness, and focality of the evoked percepts. And finally, to kind of tie this all together, again, I did a lot of different things, but I think they're all kind of tied together under this framework of biomimetic stimulation, where biomimetic stimulation itself is not going to produce completely naturalistic sensations. It's not completely groundbreaking, or it's not a huge breakthrough in the field or something, but I do think it is actually the superior way to provide sensation. And the first reason for this is that, again, it provides less charge injection overall for the same force profile, which can potentially result in better electrode longevity, which is going to be important in these clinical applications. The biomimetic frequency modulation, as we saw in the second goal, will likely need to be conducted on a more electrode-specific basis based on the underlying somatosensory organization. So we really need to better understand the somatosensory organization and move forward on this front. And finally, the amplitude modulation was able to improve the naturalness and intuitiveness and decrease the predicted field size in one participant. But that's actually an important point. All this biomimetic work that I presented here was in one participant. And so moving forward, I think we really need to test this in multiple participants because biomimetic itself can be kind of subjective, especially how it affects naturalness. And so we want to see if this extends to multiple participants. And then also biomimetic encoding really should be tested in the context of BCI tasks. So all the work here, I was really evaluating the perceptual metrics, how biomimetic encoding can affect perception, but didn't really investigate how it affects function. So I would argue that, you know, if biomimetic encoding actually improves function, that'd be great, especially for certain tasks. But even if it resulted in similar performance to linear encoding, I think it is actually a better method in terms of, again, less charge injection and more natural qualities being evoked. But these next few points are more like future directions, I think, for the field. And likely, I think newer technologies will be needed to develop better targeted stimulation. And this is because even when we use these biomimetic methods, we're still stimulating these large swaths of neurons, and typically individual neurons might behave differently in the population, especially different types of neurons, which is something that's really not well studied right now. So the contribution of different inhibitory or excitatory neurons and how those things you might want to recruit them is going to be, you're going to need newer technology to be able to do that. And finally, a bit of plug to my own future work as a postdoc. I think that animal experiments will need to be done to really understand the underlying mechanisms of both stimulation and the organization of somatosensory cortex. And so I'm actually going to be doing 2-photon microscopy in mice in TK Kozai's lab to try to better understand these mechanisms and the organization of the cortex so we could build better algorithms for feedback. And finally, I kind of just want to go over the things that I produced from my graduate work. And so these are publications that are currently kind of out. So I have 4 first author things, 2 are research articles, and they're currently on archives and in revision at eLife and Journal of Neuroengineering. And then I have 1 conference proceeding, which was selected actually as a top 10 conference paper and for full publication in the proceedings of, is it neural systems and rehab engineering. And then a first author book chapter, which basically just covers a lot of the background for this stuff. I also was a co-author on 2 papers, 1 conference proceeding, and the recent science paper that I talked about earlier, which Charlene was first author on that got published recently. And these are the things that are still in preparation. So I have 2 first author research articles. 1 of those is fully drafted. I'm kind of just waiting for feedback from co-authors. And then the other is drafted, but we're really waiting on data, which is that biomimetic aim, because again, these kinds of things might not extend to multiple participants. So I feel like we really should do more experiments before we really try to publish these results. And then I have 3 second authorships, all of the, and 1 co-authorship. I believe all of these papers are basically in the final stages and hopefully will be submitted soon. So looking forward to getting those things out. And then finally, all the things that I presented. So I was actually very fortunate to be able to give 4 podium presentations during my graduate studies. One of these was in Japan, which is really awesome. The other 3 were supposed to be in Belgium, Montreal, and Tuscany, but they all got moved to online because of COVID. So it kind of sucks I wasn't able to go to those places, but I was also, still very privileged and to be able to talk to people about this work. And then obviously a lot of poster presentations as well came out of this. And I'd like to thank the people who are involved in this work. So in particular, our participants, Nathan and Mr. Dom. So Nathan's been implanted for a long time, and he's always been very engaged and contributed a lot to the experiments. And you know, I think he not only helped directly in my work, but also just really pushed the field forward in general. So I'm very grateful for Nathan as a participant, but also in these years, you know, I've come to know Nathan as a friend, and I appreciate him in that regard as well. So I'm very lucky to have worked with and got to meet Nathan. And then Mr. Dom's been in the study for less time, and so I collected less data and also didn't have as much time to know him, but I still considered him a friend, and he's a really awesome guy as well. And he's been continuously excited and engaged in this work. And I'm hoping that we can continue to keep him excited about the work that we're doing. And then faculty, Rob, obviously, he's my PI. None of this would be possible without him. He had to hire me for me to do this work. So, but even beyond that, you know, he's a good mentor. He provided a lot of insight into these experiments and analyses. And so we wouldn't have this end product if it wasn't for Rob. And then both Jen and Sleeman are close collaborators on these projects. So Jen more so on the BCI side and Sleeman on the sensory side, particularly. I mean, I brought up Sleeman's name a lot throughout the presentation because in terms of the ideation as well as the development of these biomimetic trains, he was very important. And they're both on my committee as well because of how closely we work with them. So I'm really grateful to them for their contributions both to the science and on my panel. And then Aaron Batista wasn't directly involved in this work that I presented here, but he is on my panel and actually, I think, helped probably the most with developing my dissertation because I had a lot of confusion about what I should include or should not include, and Aaron kind of helped me streamline that. So I'm grateful for him as well. And then Mike Boninger, who's not on my committee, but was really pivotal to getting a lot of this work started and really getting it, and really still to this day, forming the ideas and moving it forward. And then there's a lot of other contributors in terms of students and engineers and staff, including, I want to highlight just three. Charlene, who did a lot of the previous work that really led to the motivation of some of the things that I did. And then Jeff, who's an engineer in the lab, helped me really earlier on figure out the system so I could start modifying and building my own experiments. And Dave helped particularly, another engineer in the lab, helped particularly with developing a new voltage monitor, as well as the mechanical indenture that was necessary for some of those biomimetic experiments. And then also people in Tracy Schwee's lab who contributed to this material stability stuff that, again, hopefully will get submitted soon. And then on a more personal note, I'd like to thank my fiance, Madeline, who's pictured here on the left. So she's provided a lot of moral support these last few years, and I'm looking forward to the many years we'll spend together, even after I hopefully pass or get my PhD. And then obviously, or not obviously, but I made a lot of friends throughout this time too that are pictured here on the right. And as rich as my career was in science, I think it was also rich in friendship. And I'm appreciative for all those people who I've gotten to know over the years and who've been there. So thank you. And with that, I'll end and open up for questions. Thank you.
Thanks, Chris. That was a tour de force. Really incredible job. Way to go. I'm really proud of the work that you've done and you for doing it all. Questions, again, committee, if you can hold your questions, but anybody feel free to just pipe up. I'll keep an eye on the chat too if you want to post messages there, but otherwise feel free to speak up and ask Chris those questions that are most on your mind.
I have a question.
Go ahead, Lee.
First of all, really nice job, Chris. I thought you did a good job with all the work, and the talk was great too. You know, in thinking about the frequency preference stuff and also the biomimetic approach. So, you know, the fact that you get this frequency preference, I think that every stimulus pulse probably generates an action potential in the neurons that you're stimulating, right? Like if you're doing constant amplitude stimulation. So that suggests to me that it may be some processing downstream from those neurons that's actually where the frequency preference is actually being exhibited, right? Because those neurons are just firing at whatever rate you're stimulating, I think. Where do you think that's happening? Like, what do you think is actually going on here that results in that frequency preference?
Yeah, so there are a lot of ideas about potential mechanisms, and I can kind of go through all of them, which I'll kind of summarize some of them. But basically, this is something, you know, that's very difficult to study in humans, which is why I want to do this postdoc, where I'm going to look at these mechanisms directly, right? But the one sort of idea that I really strongly believe in, and it's sort of what I hope to start doing in my postdoc, is that there's sort of different distributions of different neuron types in the cortex, right? So there's these different excitatory and inhibitory neuron types. Particularly, there's these like parvalbumin and somatostatin neurons that could be regulating the activity. And so even though you're recruiting all the neurons in that distance with sort of the same amplitude and same rate, the way those neurons are actually interacting in the network is going to be different based on how they're connected and what their identity is, right? So these somatostatin neurons, for example, can actually regulate both parvalbumin and excitatory neurons. And if those neurons are actually able to entrain to different frequencies, it can affect the way it's regulating other neurons. And the somatostatin neurons are actually less prevalent too in the somatosensory cortex. So if you happen to be in an area that has more somatostatin representation, you could get different kinds of responses. So this is something that's not very well understood, but it's kind of an idea in my mind of what might be going on. But this also could be due to projections to downstream areas, as you mentioned, which also could then be related to the organization of those areas. And in fact, with the 2-photon microscopy, we're going to be looking more at layers 2 and 3 because of the recording depth. And so the idea is projections to those areas from layer 4, which we believe we are stimulating, can actually impact these relationships. Some other ideas too, though. So I'm mentioning sort of the presence of these different inhibitory and excitatory cells. But even on another basis, you can have this sort of habituation of individual neurons based on their ability to replenish neurotransmitter. And this has actually been looked at before, I think, in some computational literature as a possible mechanism for frequency filtering in the brain. So there's, and then also, you know, where our Utah rays are hopefully level with the cortex, but they can also be tilted. And so you might also be seeing some layer specific effects of the neurons that you're directly recruiting. And again, the way these neurons project to other areas might affect the way the perception's ultimately shaped. But I just think, so again, I thought about this a lot and I have a lot of ideas of how it might work, but we can't really answer these questions in humans, which is why I'm really looking forward to doing some of this work in animals to try to answer what's going on.
Chris, I have a question. Great presentation, by the way. So, you know, of course, I work on the periphery, and like in the periphery, we have this concept of afferent noise, right? So, and you're trying to mimic that with your biomimetic stimulation. So, have you ever wondered that noise might play a role that is, you know, probably currently beyond our understanding of like designing that pulse and trying to again push that back into a cell?
Yeah, so it's an interesting question, because I would say with the biomimetic stimulation we were providing, we weren't necessarily trying to reproduce this sort of neural noise, because the shape of the trains typically have this characteristic sort of onset offset, which wasn't really necessarily representing noise, right? But I definitely, like, I've read a paper in a computational neuroscience journal where they looked at actually how noise itself can provide sort of optimal encoding in the cortex, and actually noise can help improve information transfer. So noise itself might actually be relevant, but that noise in the cortex, typically we see it occurring on a more neuron specific basis. And again, we can't really recruit neurons specifically with our method of stimulation, right? So I think we're trying to really reproduce more of these population level processes. And so reproducing this noise, I don't necessarily see it being as useful in this context, but I think noise itself potentially is important for information transfer. I know there's been some work, or at least some ideation on using this in the peripheral nerve system, and it would be interesting to look at it in the cortex, but I think based on these previous results we've seen where we do sort of timing specific modulation, we're going to see similar results with this sort of introduction of noise. But I think it's still something that'd be interesting to look at.
Thanks, Chris. Great work.
Thank you.
Questions from anybody else?
Can I ask another one?
Fine. Yes, go ahead.
Sweet. In your AIM-1 stuff where you saw the thresholds going down.
Yep.
Did you look to see what? So there's kind of two, maybe, and probably other potential explanations for that. One is that the thresholds were actually going down, and two is that the high threshold neurons were going away, or the high threshold channels were going away, right? Because you had a decrease in the number of channels as well that were evoking a response, I think, or was that just the recordings?
Yeah, right. So we just saw decreases in recording. Actually, the amount of electrodes we were able to evoke sensations from went up over time, but sort of leveled out. I want to say around day post implant, maybe 300 or so. And then we were basically able to evoke sensations on almost every electrode. But then we kind of had this other effect where we started to see higher voltage transients on electrodes. And if we have high voltage transients, we disable those electrodes because we don't want to cause potential material damage by stimulating at lower voltages. But even that was kind of an insignificant amount. We were basically going from 60 to 50. But from day 100, where we saw the highest thresholds, the number of electrodes actually only increased in subsequent test sessions.
Okay, and if you look at that subset of electrodes where you had responses at whatever, day 100 or whatever, and then you look at those same electrodes 2 years later or something, you saw decreases in thresholds like on a per electrode basis there, as opposed to just like looking at the means.
Yeah, I don't know if I included this figure. I have a bunch of backup slides. I don't think I actually included that figure, but there's a figure in the meta archive paper where you can actually see heat maps of the individual electrodes, and where basically I think it's like brighter colors represent higher thresholds and cooler colors represent lower thresholds. And you'll see the individual electrodes themselves are actually changing. So it's not that you're getting more electrodes with lower thresholds. The electrodes themselves are actually, the thresholds are going lower over time.
Okay, cool, thanks.
Chris,
Hey, Steve.
to follow up on that, I was wondering, because I think the results from your M1 were particularly interesting. Do you think that just the delivery of the stimulation itself is providing some sort of therapeutic effect that's why you're retaining more units, or you're seeing that these thresholds are becoming lower?
Yeah, so that's really hard to say. That's kind of, it'd be a really cool result if that was the case. But there are also other substantial differences between the electrodes, right? So just in terms of the material and the implant location. And in fact, we're now, that paper's in revision, and we were asked by a reviewer to look at how the actual amount of charge impacts these relationships. And I've looked at it, and we see that the amount of injected charge doesn't affect the peak-to-peak voltage. And it also doesn't have a significant effect on the high amplitude waveforms, although there is a slightly positive slope where more charge was associated with better waveforms. But this is very preliminary. I actually just started looking at this recently. And there also seems to be, again, a not quite significant, but almost significant effect on the impedances, where more stimulation led to lower impedances. And lower impedances could mean better recording quality or better ability to record high amplitude signals. So that might be a potential mechanism by which stimulation is actually improving our ability to record signals. But I think it's really difficult to say without looking in arrays that are more equal in terms of the material. So I think that's, our current participant P3 has iridium oxide on both arrays. And so it'll be interesting to see how they compare in him, since we're not currently at least stimulating on these motor arrays.
Yeah, those are good points. So when you explant these arrays, you look at the material like characterization, but do you also do or does Tracy Lab also do any sort of immunoreactivity to see tissue response on those arrays, and if they differ between the non-stimulating ones?
Yeah, so they did a bit of that analysis, which I didn't include here. There's actually a lot of stuff in that manuscript that I didn't include here. I just kind of want to focus in on the stimulation, on the material integrity. And I can't really speak to it as an expert myself. But also the arrays that were explanted, there were sort of a lot of complications where we only had 2 stimulated arrays, and they actually had different, one of them was encapsulated and one of them wasn't encapsulated. And then the motor cortex arrays had a similar behavior where one of them was encapsulated and one of them wasn't. And then they were across different participants. And we also explanted this first set of arrays from P2 months after the pedestal was disconnected. So there's a lot of issues with actually associating the data and the material integrity closely. But I don't think, as far as I know, I didn't think there's any significant result in terms of the stimulation and immune response. Because I believe they looked at that, but I think it might be like a supplemental figure. So on the top of my head, I can't give you details. But
okay, that's fine. Yeah, I just think it's interesting to think about. Thank you.
Mm-hmm. Yeah, so I guess look forward to that paper getting submitted.
Right, it's 3 o'clock, but if there's maybe one more question, if anyone has something they want to ask quickly.
And also, if people do have questions and don't want to say them out loud for whatever reason, definitely feel free to email me. I think my emails are.
There's actually one in the chat from Joel. It says the stability analysis where you showed electrode degradation might not imply signal degradation. That was studied with implanted electrodes, right? Yes. Is this something we studied ex vivo? I don't know, Joel, if you want to speak up and say anything about that, or.
The question basically summarized it.
Okay.
Yeah, I was just curious because it seems like that particular result could be something that could have been looked at, like just looking at the electrodes themselves and like simulating wear and tear or something like that.
Yeah,
I don't know,
Yeah, I think it's kind of interesting because there's been a lot of studies, I mean, in Tracy Shui's lab and other labs, where they looked at how stimulation affects the electrodes. And it almost always seems to be a negative impact, right? Where you're getting material degradation. But again, so this is why it's so surprising, I think, on these implanted electrodes that we're able to continue to record and stimulate for very long periods of time. And we've talked to Tracy about this a bit, and we have some ideas of why this might be happening. So it's possible that those long-term implants actually still do have material damage, but because they're in the brain and the metal stays intact with the electrode due to that sort of environment, we can actually continue to do things with these electrodes. So I think, again, yeah, I think doing more experiments ex vivo maybe could help clarify some of these things. But I also think that based on the ex vivo experiments, there were sort of predictions that maybe aren't holding true with these implanted arrays. So I think, sometimes taking the ex vivo to the in vivo can be a bit difficult. So.
Okay, I gotcha. Thank you. Wonderful presentation.
Thank you.
All right. With that, I think we will wrap up. Thanks again, Chris, and thank you to everybody who called in and listened to this and showed your support. Sleeman, Aaron, and Jen, if you're able to stay on, that'd be great. But for the rest of you, thanks again for calling in.